Hello and welcome to my channel. I would like to discuss today quaternary structure. This is the fourth level of structural organization which is present in a folded protein. You have learned that the primary structure undergoes local folding, forming secondary structures which together undergo further folding, leading to the formation of a compact 3D or native conformation known as tertiary structure. In many protein you will see more than two polypeptide. Two or more than two polypeptides are required for its activity. These polypeptide must be in their tertiary structure. So this type of multimeric structure of protein is known as quaternary structure. In this video we will explore what quaternary structure is in detail, what are different forces that are responsible for their formation, and lastly the significance of quaternary structure in human health and disease. So what is a quaternary structure? Quaternary structure is an association of two or more polypeptide chains in their tertiary structure. Many proteins, they require this association for their functionality, for their activity. These polypeptides are also known as subunits. These subunits may be homomeric or heteromeric, like homodimeric, heterodimeric, or heterotetrameric, like in case of hemoglobin. So basically these are stabilized. These subunits are held together with various type of bonds which we are going to discuss. So the interaction, these forces are between the subunits. This is very important point to note here. So what are the example of quaternary structures, or you can say molecular teamwork or this association? The first example is collagen, which is homotrimeric. There are three chains, similar chains are present in the collagen molecule. The next example, hemoglobin, which is a heterotetrameric protein. It has two alpha chains and two beta chains. So there are total four subunits. This is a good example of heterotetrameric. I will give you an example of multimeric, heteromultimeric protein such as pyruvate dehydrogenase complex. It has 60 subunits. Why a protein has to have multi subunits or multi subunit structure? This is for providing better regulation of an enzymatic reaction or metabolic pathway in which they are functioning. So this is the significance of this structure. Now coming to the stabilizing forces, the first point you should note here is that these interactions are between subunit. So this is an example of interchain interactions or bonding. And what are the group? These are again side chain groups of the amino acid residues in present in different subunits of a protein. So first one is ionic interaction. It is electrostatic interaction between negatively charged and positively charged side chain group present on the amino acid in two different subunits. So this is ionic interaction. The participating amino acid could be aspartate or glutamate. The other one may be lysine or arginine. So acidic and basic amino acid residues normally participate in this type of interaction. The second is hydrogen bonding. Here also hydrogen bonding is functional, and this is between two subunits. One is providing OH group, other is providing the carbonyl oxygen. So carbonyl oxygen comes from acidic amino acid residue such as aspartate and glutamate, and H is here from serine. So this is also, as I mentioned earlier, it is a common type of force that stabilizes all type of the protein structures. The next one is hydrophobic interaction. As I mentioned again, these amino acid have a tendency, this is the tendency of hydrophobic amino acid side chain groups to make to hide from or to cluster together away from the aqueous environment, and this is a major force that helps a protein to acquire final folded structure or quaternary structure. The last one is very important, a disulfide bridge. I have marked it as occasional. The reason is because this is not present very commonly in quaternary structure. It may be present but not commonly present, so it is rare. And this is as usual, this is between two cysteine residues present on two different subunits. So this is called as disulfide bridge. The reason for that is it could be because of the labile nature of SH group in cysteine. They are highly labile and they might be consumed by the time a protein acquires tertiary structure. This is hypothetical explanation, so you should know that. So this is all about the forces, different type of forces that stabilize this structure, quaternary. So what is the significance of quaternary structure? Now you must keep in mind that any change in the primary structure of protein will be reflected similarly in the tertiary and quaternary structure. An example is sickle cell disease, which is caused due to the replacement of glutamate 6 by a valine amino acid. This is the normal structure of hemoglobin. In beta globin at position 6 there is a glutamate. However, in sickle cell disease this glutamate is replaced by valine 6 and this causes formation of hemoglobin S or sickle cell hemoglobin, which acquires a fibrous structure leading to distort the overall structure of RBCs, which resembles like sickle shape. So this disease is called as sickle cell anemia. So this is a good example of defect in the quaternary structure leading to a disease pathogenesis, sickle cell anemia. So this is all about the significance. There are many other examples you can cite, but this is good enough for you to know. So finally, thank you very much for listening. I would like to encourage you to watch the complete video so that you get full benefit in your examination. Please like it, subscribe it, and share with your friends who are interested in learning protein biochemistry. Until next time, take care and bye bye.